All right, A&P2 students, we're moving on down to, we talked about the structures that are part of the upper gastrointestinal tract. It kind of starts with the oral cavity, pharynx, esophagus, stomach. First part of the duodenum is kind of still considered upper gastrointestinal tract. But now we're moving into the lower gastrointestinal tract, which will be the small intestines and the large intestines, and we'll talk about the accessory digestive organs too. But first we're going to focus on our small intestines and the information that we need to know about them. This is the objective 20 that talks about the functions of the small intestines. So chemical digestion is going to be occurring in the small intestines. That's where we'll have enzymes breaking down our nutrients into smaller molecules. Absorption is a huge function of the small intestines. That's the reason why the small intestines are very long and why they have, if you remember from lab, we have circular folds. That gives the increase in surface area for the small intestines. We have villi, that increases surface area, and then we even have the microvilli. So we have three things that are going to increase the surface area of those small intestines so that there's plenty of areas where we can get absorption of nutrients. So we're going to mainly absorb all of the nutrients, a large amount of the water and the electrolytes too. So most absorption goes on in the small intestines. All right, we're also going to get mixing. Remember they call it segmentation. That chyme that's coming from the stomach, it needs to be mixed with the fluid that's coming from the accessory glands, which would be like the pancreas and the gallbladder and liver. So we need to have that mixing going on so that all those secretions get in contact with all the parts of the chyme, all the nutrients that are in there. And then along with the motility, 1-3 is peristalsis. So once all that mixing's done and we've got digestion going on and absorption, anything that we're not going to utilize, it needs to be propelled down towards the large intestines. So those peristaltic movements that we saw, contractions that we saw, are going to happen along the small intestines to move everything along that we're not going to need to use. All right, and then when it comes to chemical digestion, the secretion of enzymes needs to happen so that they can chemically digest certain nutrients in the chyme. And so secretion of enzymes is a function of the small intestines. I have 4 of our enzymes that are in our small intestines. Obviously there's a lot more, but these are the 4 that you should be familiar with and know what they do. So lactase, they all end in ASE because they're enzymes. So lactase is going to digest the sugar lactose. And then sucrase is going to digest the sugar called sucrose. Peptidase enzymes, that's a pretty general term for them, but there's different types of peptidase enzymes, but all of them are digesting some type of protein. And then the nuclease enzymes will digest DNA and RNA, or any of the nucleic acids. So, all right. So let me remind you the role of enzymes here, because we're going to be, so I made this very simple picture, because I can't draw on the board, so I use this paint program. So I'm showing lactose. So this is the molecule lactose. I've drawn it here. So it has these 2 sugar molecules. One's a glucose and one's a galactose, and they're bonded together. So they have their little bond here. And so we get the lactose from milk. So when we have, consume milk or any other dairy product, we're going to have this molecule lactose in our small intestines. And so, unfortunately, this is too big. It's only 2 sugar molecules, but it's too big to be absorbed. So we have to break it down. Our body's got to have something that'll break it down so that we can get these little sugar molecules separate from each other. So they'll be smaller molecules, and then those can pass through the epithelial cells and into the blood. So that's where the enzymes that the small intestines that they secrete And so the enzyme and enzymes, remember, are specific for their substrate. So this is the only enzyme that's going to break the bond in this particular substrate or lactose molecule. So lactase is the enzyme. I made it a little blue triangle. So it's going to come up here and it's going to break that bond. And once that happens, then we'll get individual sugar molecules. Now those guys are just the right size to pass across the small intestinal epithelial cell and then get into the bloodstream that's in the lamina propria part. So that's how it's going to go for, I just used that one as an example, but that's how it's going to go for all these DNA, RNA, proteins. All of these are too large of molecules to be absorbed, so we have to have enzymes that are going to break them down into smaller molecules so they can get across and be absorbed and we can use them. All right, and then the last function of the small intestine is hormone release. We talked about it up here when we talked about the regulation of the stomach. So I added that. I actually added this, so I'll update these on Blackboard. But remember that the small intestine, there's cells primarily in the duodenum area that are releasing these hormones. So cholecystokinin and secretin are two hormones that are being released, and they're going to regulate the functions of the stomach and the intestines too. So, all right. So what I'm going to do is, since the next thing we're going to talk about before we talk about the large intestines is we're going to talk about the accessory gland organs, so the liver, gallbladder, and pancreas. And the reason we talk about them now is because their secretions are going into the small intestine, specifically into the duodenum through that major duodenal papillae. So we want to talk about how those affect digestion in our small intestines. So I will start another video which will talk about the accessory gland organs and how they function.